Welcome back to the lecture about protein function prediction, and this time we're going to talk about mainly docking and protein binding site comparison. So last time I said that the prediction part of what is the function comes largely from the observation that similar proteins have similar interaction partners. We talked about binding sites and what you can do to analyze a binding site. This time it's going to be how to superimpose two binding sites and find some that have similar properties. And we're going to look at how can I see whether a specific ligand binds into a binding site. Okay, what do I have to do to compare two binding sites? As I mentioned before, you first have to find the superimpositioning in 3D. And in a way, first finding the superimposition or the matching in 3D when you want to compare them is a kind of hen and egg problem, because then if you can find how they superimpose in 3D, you already know what is equivalent. So it's trivial in cases where you have sequence similarity or structural similarity, because that gives you the equivalence. Okay, then I said the really interesting problem is when you don't have sequence similarity or structural similarity. An example for two proteins that bind something similar but are not related sequence-wise are those two. One is a kinase and one is a reductase, so they do some pretty different things. What they have in common is that they bind a molecule that has a common part. And you would expect, or you could think, that the binding site should look similar for those parts, because they have to do the same kinds of interactions, and you have developed a successful method if it can find those two proteins to be similar. Of course, there are also other examples, which is the one that is mentioned in the paper, so I'm taking that one. If you look at a 2D plot of the interaction between the respective ligands and the proteins, it at first looks pretty dissimilar. And you'd say, no, they don't have anything in common. Now, if you blend out the stuff that doesn't belong to the part of the ligand that is similar, you start being able to find the similarities. For example, what is pretty nice to find, for example, is that these NO, you have a H bond there and the NN too. And you find the same thing with NO and NN in the other binding site, only in this one you do it with two different amino acids, which are not really neighboring to each other. And in this case, they come from the backbone of the same amino acid. But they do make the same kind of interactions. So that's our goal. We want to be able to find that. And the method I'm going to show you in a moment in more detail does this by representing the binding site with pseudocenters, which tell you something about the type of interaction the binding site can make, and then matching those pseudocenters onto each other, and then in the end you see that the ligands can be superimposed and the binding sites can be superimposed. Okay, so what are the things I have to do if I develop a method to find this kind of match? Well, first I need to be able to automatically locate the binding site, because I want to do it over a whole database of structures, and I can't define that by hand. The next thing is I need to be able to somehow represent the important features in a binding site, the stuff that makes the binding site bind the ligand. And then I have to be able to search efficiently against over the whole database. And the searching is finding a good superimposition and also scoring that, finding those matches that are significant. Okay, there a number of methods exist. I'm going to show you two. The first one was, as far as I know, the first one that did it. But I'm also biased towards it because it was developed in the group where I was doing my PhD at the time when I was doing my PhD. So it was a colleague, and I of course love to present that. And the other one is one that is a lot faster and can go can do whole database against database searches. But the point why I'm presenting this is just for you to get the concepts behind it. The representation of a binding site can be either directly by the atoms. The drawback if you take the atoms is that they're loads. So therefore the methods I'm going to show you don't really take the atom positions, but they abstract a little from them. You can take chemical groups, where you can kind of classify by type. Say, okay, this is a H1 donor, a H1 acceptor, things like that. And you can take surface points. And another thing is to have some very abstract shape descriptors. The way to superimpose is usually some kind of geometric hashing or clique searching. And this graph theory and clique searching, we are going to see that again and again in this lecture, and I'm going to explain in more detail what I mean there. Similarity measurements are usually done by the RMSD or by residue conservation, which is of course easy to calculate. You can just take the substitution tables, or you can look in more detail and look at the physicochemical properties. Okay, so as I mentioned, CavBase was as far as I know, the first one to do this binding site comparison. The cavity detection was done with LickSite, which I already showed last time, with the grid, filling the grid and finding the buried points. The representation of the binding site is done by going through so once you have found the binding site, you go around and see which residues do I find there and what kind of interaction points do I find in the binding site. And it's classified as hydrogen bond donor, hydrogen bond acceptor, something that can be both. Do you know what kind of functional group can be acceptor and donor? Anyone? Can anyone tell me any hydrogen bond donor? The thing it gives a bit, yeah.
Acidic amino acids?
Yeah, but well, I'm talking about hydrogen bond. Yeah, well, okay. If it's got the protein attached, then it can be a hydrogen bond donor. Yeah. Once the protein is gone, it can't give it anymore. And hydrogen bond acceptor. Which atoms would be hydrogen bond acceptors? Yeah.
Oxygen.
Right. And what would be a typical functional group where you would see that? Like, the oxygen doesn't exist by itself, but it's usually attached to something. Yeah.
CEO, like with the bond.
Yeah. And yeah, I've got one here. So this CO, and here you've got the free electron pairs, and with those free electron pairs, it's a hydrogen bond acceptor. Okay. So now, what could be both? Which functional group? Yeah.
Yeah, OH.
Right, that's what I was talking about. So an OH can be an acceptor in the free electrons, and it can also give its H. And therefore, you also have this type donor acceptor. Then you have the type aliphatic, because this can make like hydrophobic interactions, and a special type pi, because these can make this kind of pi stacking. And of course, per residue, you can have several of these types. Okay, so then you represent every of those groups which you find by a pseudocenter. And here in the picture, the color says its type. Like this would be, the red would be hydrogen bond acceptor, blue would be hydrogen bond donor, etc. Okay. Apart from representing them by those pseudocenters, they also mapped the properties to the surface. And you will see later why this is done. For now, just remember that this is used for scoring, for seeing how well two binding sites fit each other. Okay. Now, for every binding site, every single binding site, you can create a graph that represents that binding site. So that's an abstract representation. And a node in the graph is any of the pseudocenters. And two of those nodes are connected by an edge. And the edge is kind of annotated with the distance between the pseudocenters. So for every pseudocenter, you can go through the list of all the others and measure how far is this apart from any of the other pseudocenters. And then you store in your computer like I have this pseudocenter and it's that far apart from that other one. Okay, that makes a graph. Now, if you want to map two binding sites onto each other, you need to find the equivalencies. And as I said, finding equivalencies is trivial. So it would be trivial if you had the 3D similarity of the overall structure, but here we have to get it just out of the binding site. So we have to find out what is actually equivalent between the different pseudocenters. So what we do is we make this associated graph. We take a pair of nodes from protein A and protein B. Like for example, we have node 1 from A and node X from B. And we look at, is it the same type of pseudocenter? If it's a donor in both cases, that's fine. If it's an acceptor in both cases, that's fine. So this can give a node in the associated graph. So X1 can make a node. And here we have 2 and Y. And 2Y, let's say, also are the same type, so they can make a node. Now we need to say to look, can we have an edge between them? An edge between, what do you think an edge between them in the associated graph would mean, like logically? What would be why would you want to make an edge? Yeah.
You'd make an edge if they correspond.
Right, if they are compatible, if matching X and 1 is compatible with matching 2 and Y. And under what condition is it compatible? Yeah.
If the matching patterns are the same?
Right, and matching pattern in that case is first the distance. So if the distance is too different, then you can't superimpose this to that and that to that. So what you do in order to look, can I make an edge here, is to compare those distances. And that's what it says on the slide. You look at the distance difference. And the other thing is that you just observe that if you go to large distances, you get swamped with things that are not really relevant. And therefore, if the memory node distance is above 12 angstrom, you just say, okay, I don't care about this.
So it's only pairs every time?
Yeah, you just make pairs, but then of course you extend this. Now you say, okay, I have another pair, and I see whether I can connect it to 2Y and whether I can connect it to X1, etc. Yeah?
What's the member node distance?
So the member nodes of this one, so this node, which I call X1, the member nodes of that come from the individual graphs, and the member node would be X, and the member nodes are X and Y and 1 and 2.
So both have to be below 12?
Yeah, right.
Oh.
Both have to be below 12. Okay, so then I create a complete graph of this, and then I can look for the maximal common subgraph. So what is connected in this whole thing? And this gives me the set of pairs which I need to superimpose in order to find the superimposition. Okay, and this kind of thing is found again and again in like alignments and also in matching ligands to binding sites. So this is something that's worthwhile remembering because it reoccurs again and again. Okay, then this finds you a way to superimpose. Now you still need to score. And the way to score is to look at how much of the surface really matches, because the surface, so the idea behind this is that the surface is the thing that really interacts with the ligand. And so basically the surface is represented by some patches, and you just look at the geometric overlap of similar type surface patches. And the thing that they still had to somehow solve was the problem that a convex and a concave surface first looked the same, but they somehow solved that. Okay, so that was CavBase, and the test that's referred in the paper is that they tried to look for the example I introduced in the beginning. They took the kinase and searched against all of the PDB in order to reduce computation time, and because it's trivial anyway, they threw out anything with a high sequence identity. Well, I don't know what the cutoff was. And they found the first 8 ranks, they found cavities which have a similar fold. You didn't get them by the sequence identity, but if you looked at SCOP, FSSP, or whatever databases, you found, okay, they've got the same fold. So in a way, it's not really interesting. And on the 9th rank, they found the other protein I had mentioned, the reductase, which indeed does bind a similar ligand part, and so they said, okay, our method is successful, and they later applied it to other things. The drawback behind the method is, I mean, it ran on computers of around 2000, so the computers then were of course slower than now, but at that time it took on the order of days to scan against the whole data, to scan one test structure against the whole database. So if you want to do an all against all, that's of course too slow. I don't know whether anyone has since taken that up and made it more fast. I haven't seen any papers on that. A different algorithm, which is newer, it's called SWIPA. I always forget what the exactly stands for. So what they do different is they abstract a lot more. So in CavBase, which was done by chemists, they really try to go to the exact details of how things interact. Here they say, okay, we want to be quick, and we just go for amino acid type. What they do is to first look at the overall structure and represent it by the C alpha positions. Then they fill the space with tetrahedra around each C alpha, and they define 2 boundaries. One is an outer boundary, one is an inner boundary, and that with these 2 boundaries, they're able to the difference between these 2 boundaries gives you the binding site. So the outer boundary is this one, and the inner boundary is that one, and the difference between those gives you where the binding site is. Then they take every C alpha atom and make a vector that points to the boundary. This gives you a direction, like where. And in addition to that, they store for every of those C alphas the type of the amino acid. And again, they make a graph. Each node is one C alpha atom, and they make an edge in the first graph if the two tetrahedra are connected to each other, so they touch. So only adjacent amino acids get an edge. Okay, then they make again the associated graph in order to find the things that can match. And there they say, okay, node A and node B need to have something called a similar geometric potential. The geometric potential is this vector to the surface. So they kind of need to point in the same direction. And each node also gets a weight, and the weight comes from the substitution table. So if it's the same amino acid, it would get a high weight. If it's a similar one, because both are acidic, etc., that will also get a high weight, and something that you wouldn't substitute for each other gets a low weight. And we assume that if it's unlikely to be substituted, then is also unlikely to make the same kind of interaction. Now you get an edge in the associated graph if the individual member nodes here are connected. So we don't really calculate the distance yet, but we just say, okay, those two in protein A and those two in protein B are neighboring. Now we also start to look at the distance difference and see whether this is below 2 angstrom. And we look whether the vectors to the surface are also similar. And now we don't just have to find a maximum subgraph, but also a maximum weight subgraph, because we want to see that the match is good. So we kind of do the scoring while looking for the subgraph. And the score is calculated from the substitution table, the distance, and the angles. And the special thing about this Schafferhans method is that they also devised a way to make the scores comparable across binding sites. So you can kind of have an e-value, p-value, like if you did a blast search. This is pretty cool about this. And in order to do that, they had to think about, okay, what's my background? What do I compare against? And they took some unrelated structures and put on random sequences, and then looked at what kind of scores do I get for that, and then I approximated that with an extreme value distribution. And now if they have a certain score, they can compare it to this randomness and say, okay, this is significant or not. Okay, an application of this method. So they publish lots of papers with lots of different applications, but I think they're pretty interesting. So one thing they try to do is look for why do estrogen receptor modulators have side effects. Estrogen receptor modulators are used for treating breast cancer, because this is often caused by estrogen receptor being overactive. And in order to find what kind of side effects do these things have, they thought, okay, we're going to look for binding sites that are similar to the binding site of the estrogen receptor. Because a lot of the sequences that might be relevant have not been really studied in 3D. They first had to map the sequences to structures. So they didn't really build homology models, but they at least assigned 3D coordinates to every C alpha. They limited their search to druggable proteins in order to not have too much computation time. Then they searched against this database with their method, and they found on one of the first hits, or maybe even the first one, protein called sarcoplasmatic reticulum, ATPase. And if you look at what this thing does, then you see that it's involved in muscle movement, for example. And the side effects that you see in AR are, for example, cardiac abnormalities. So this is a transmitter, and the cardiac abnormalities can be caused by too much activity there. So you find, okay, this really does fit the side effects. And then they also compared, do we believe this can be similar, and superimposed and showed the different binding sites, and you see, okay, the form is vaguely similar, and also if you map the electrostatic potential, this looks a bit similar. Okay, and in order to validate the whole thing, they also ran it the other way around. They said, okay, if I'm able to find this circa by looking for something similar to estrogen receptor, then it should also be possible to find it the other way around. So they took circa and searched with that one. They took two different subsites of that. So it's a larger binding site, and they took two different parts. And for each of them, they looked against the whole database, and ER alpha came at a rather top score in both cases. So that's how, okay, this is probably really significant. Okay, another example of how you can use this kind of searching for similar pockets was they wanted to have a new drug against tuberculosis. But they didn't want to kind of go through the whole process of looking whether the drug is non-toxic, etc. It always takes a lot of time to get a new drug approved. So the idea was, well, there are lots of drugs there. Maybe one of the known drugs can be used against tuberculosis, and I can just take it off the shelf. So they looked for, okay, what is a typical target I try to attack in tuberculosis? What kind of protein do I have? And this one is called NHA, and it's somehow responsible for lipid metabolism. But I forgot what the name exactly means. Well, whatever. But this thing has a cofactor which includes an adenine. And they thought, well, maybe if I find other proteins with the same kind of cofactor, I'm also going to find something that, a drug that binds there. So they searched against lots of other structures binding adenine and compared this with their tool, and they found another protein which also has a known inhibitor. Now then they superimposed the two structures, the known cofactors, and then they looked, can my inhibitor for the protein I found also fit into the pocket of the drug target? And I said, okay, this does fit in there too. And if you then really try out that compound against tuberculosis, you find that indeed it can slow down the growth of tuberculosis. Okay, so this was finding binding sites. And now we switch to the docking topic. Now I want to see how can a ligand fit into a pocket. This whole thing comes under the term of function prediction. You can try to also dock in order to find out what kind of, if you have an unknown protein, what kind of ligand could dock into my pocket. What docking is used for a lot more is to find out what kind of drug can bind to my protein. So I want to optimize the drug. So this is what I'm going to talk more about. But in theory, you can use docking also to find a ligand. If you have an orphan receptor, for example, you can try and and screen loads of natural compounds and see which one would fit in there. Okay. Okay. What do I need to do if I want to dock? Well, the problems I have to solve is I need to be able to score to find out is the docking orientation I have found significant, and if I have different ligands, which one is going to bind better or worse. So I need to be able to calculate a value for that. Of course, I first have to find the pose. How does it fit in there anyway? And what I'm going to show is how you approaches how you do that. I'm going to show a few example programs and then say something about the performance, how good are we with that, and end with an example where it's applied. Okay, just to say something about scoring and predicting things. If you try to score a ligand by any interactions it makes, your assumption is that you can predict that from things you observe in that interaction. This is just kind of reality check. If you say, I want to see how is the correlation between the strength of binding with the number of hydrogen bonds, well, there is a correlation, but not too strong. And also, if I want to see how is it with the buried lipophilic surface area, there is a correlation, but also not too strong. So this is kind of the problem we're dealing with. In general, if you want to design a compound that is a good ligand, then there are a few rule of thumbs you can try to go by. One is it helps to have lipophilic contacts because that means that you're burying a hydrophobic surface, and close to a hydrophobic surface the water always has to have a more ordered structure than otherwise, because it has to try to make the hydrogen bonds in the network that goes that looks away from the surface. So it's always more ordered there, and then if you release that, they are more free to move. Then whether having hydrogen bonds in your ligand helps or not is a bit unpredictable. So you can't really say that the more hydrogen bonds you make, the better it is, because this can also be cancelled out by the desolvation. I mean, if the protein is in solution, it's surrounded by water. If the ligand is in solution, it's surrounded by water. And so it can make hydrogen bonds in the medium instead of making a hydrogen bond with a receptor. On the other hand, it does help to have some oriented interactions. I mean, H-bond is always more oriented than just having something hydrophobic. And the general thing is, if you have a polar atom in the protein, you of course shouldn't bury it with something hydrophobic, because that is definitely going to be suboptimal. And finally, if you can design your ligand in a way that it has less rotatable bonds and is still stable and still good to take up through the gut, etc., then it's usually stronger because you have less entropy loss on binding. So if you try to design a strong ligand, these are rules of thumb you can try to observe to make it better. Okay, so if I want to calculate how the bound state looks like and what, and if I want to, or what I want to do is that I would like to be able to distinguish likely and unlikely conformations. So this is for, I have different docking solutions and I want to pick the one that is correct. And the other thing I want to do is to be able to distinguish different ligands and see whether A or B binds better. These are a bit different tasks, but usually my scoring function has to be able to do both. And the assumption I have in all this is that my bound state is in the minimum. So when I try to find the optimal docking solution, I usually assume that this docking solution is in an energy minimum. This is not necessarily true, because you could also have something where you have some freedom of movement and you can't really predict which exactly is the correct solution. Okay. Yeah, this is just to say that apart from the interactions between ligand and protein, you of course also have to keep in mind that within a ligand and within a protein you also have energies to observe. So this is just the example of a very simple molecule, and depending on how the substituents stand, you have different internal energies. And you see the kind of the order of magnitude you have here. So when we try to calculate energies, then we can take molecular mechanics force fields, which you probably have heard about before, where you approximate the bonds by springs, and you assume that, basically the molecular mechanics force field is the collection of the constants you have in here. So for every type of bond, you have the optimal length and the force constant, and then you can just look up how to calculate that. So if I have a scoring function for docking, which is based on a force field, then I'm only calculating the enthalpy, and I'm not calculating the free enthalpy. That means I'm not taking care of any entropic term, because that's just not contained in the force field. And the typical force fields that are used are charm and ember. And here I'm just, as an example, giving the equations. Of course, we don't expect you to be able to write them down, but this is to show that how they are similar and how they also might be different. So in both cases, you've got this bond term for the spring, you've got the angle term, you've got this torsion term, which is pretty similar. You've of course got the non-bonded interactions, the van der Waals and the Coulomb, which you can find here. And then charm does a bit more magic about the dihedrals and has some other terms there. Have you learned anywhere where those constants come from? How do they derive them?
Physics?
Yes, you could try and go down to do some quantum mechanics and things like that and get them from there. Depends on what you mean with physics. Yeah, what they do is to try to take small molecules and observe, for example, frequencies of motions within the bonds, of torsions, etc. Observe these through spectra, and then model that and approximate it as good as one can. So in a way, these things are empirical. So they're not really derived from first principles, but just so this is the closest we get in scoring functions to trying to calculate it from the ground up. So the next scoring functions I'm going to show you come more from approximating what we observe in binding constants. And they are called empirical scoring functions. This can be a bit confusing because in reality this is not theoretical either. This is also kind of empirical. But these are called empirical because you start with binding data and try to approximate that. One example for an empirical scoring function is Autodock, and you see some of the stuff is the same as in the force fields, The Coulomb term and the Lennard-Jones term is again there. So you've got the Lennard-Jones here and you've got the Coulomb term there. The difference is that they directly try to model the hydrogen bonds. They don't look at internal energies in detail, but they look at the number of torsions and just have a torsion term for different types of torsions. They have a solvation term. And the solvation term is there for getting the change in entropy. Okay. And yeah, so examples where it's used is CavScore, CavScore, or Autodock. Another type of scoring function is a knowledge-based scoring function. Again, the terms are a bit arbitrary because the empirical scoring function is not the only one that uses empirical data, and the knowledge-based and empirical is also a bit the same. But so this is the way people generally distinguish them. In that case, it's based on crystal structures that are observed. And you don't just take protein crystal structures, but again, you also take small molecule crystal structures. And these are collected in the Cambridge structure database. And what you can do is you go back to the Boltzmann distribution. Don't know how much you hear about that in bioinformatics. Have you come across that before? Some yes, some no. Okay, so the basic observation is if you look at quantum mechanics in detail, you can derive that there is a connection between the probability of finding a system in a certain state and the energy of that state. So there is, and the Boltzmann equation tells you how this relation is. So the probability of finding states I among all states that you can imagine is given by some factors, and here you have e to the power of the energy of that state. And I just mentioned that there are other things because this is what kind of makes the background, and it's often, if you try to derive those functions, it's often a bit difficult to say what you compare against in the probabilities. But anyway, so if you want to compare or if you want to be able to calculate this, the energy of a certain state, then you can solve this equation by the energy you find in here and say, okay, it's related to the probability of finding the state. And now if I want to compare two energies, then I can compare the different probabilities and therefore make an energy difference. And there are several of these knowledge-based scoring functions. One example is drug score. And what was done for that is to take, to look at distributions of distances of any type of atom. So you take one atom, and then you look in a sphere around it in different distances, and you see, and you look at, okay, how many other atoms do I find there of any type? And then I look at specific distances, like I look only at aliphatic carbons, for example, or I only look at aromatic carbons. And then I look at that distribution. And now I compare the two distributions, and that gives me the energy function for how is the interaction between the two aromatic carbons or the two aliphatic carbons. And because this, again, doesn't account in any way for entropy, they added a term for the solvent accessible surface and looked at how much solvent accessible surface is buried in protein complexes, and also derived a function to calculate an energy from that. And this then makes the complete potential. And it turns out that applying that, and after you have parameterized this carefully, this is often a bit better able to distinguish poses than some of the other scoring functions. And because all of this is really important for designing new drugs, this is an actively researched topic because it makes drug development faster. Okay, now we have a bunch of functions which we can apply once we have a scoring, a pose. Now, how do we get there? The problem is that we have loads of degrees of freedom. If we have a ligand and a protein, then they can move in 3D in space. They can turn. Within the ligand, we have rotatable bonds, and within the protein, we also have rotatable bonds. And usually this is too much. So when the first docking programs were developed, you kind of forgot about all the internal movement in the protein and in the ligand. Nowadays, usually the ligand is seen as flexible, and it starts that the protein is also seen as flexible. But this is always an iteration, you know. You try to position the ligand in a more or less rigid pocket, and then after you've put it there, you adapt the pocket a little, and then again you adapt the ligand a little, and you adapt the pocket a little. So what are typical? So there are loads and loads and loads of docking programs, and I'm sure I don't know everyone. But you can try and classify them by the type of approach they take. A very simple approach is to just do a shape matching. Another approach is to incrementally construct the ligand in the binding site. Another thing is that you have genetic algorithms, simulated annealing, Monte Carlo, in order to find out how can I put my ligand in there. And some tools like Autodock provide you with several options of how you can optimize. And as a user, you can just choose what you like best. I'm presenting Dock because it's still pretty good, and it was one of the first, so it's worthwhile knowing. What they do is they present the active site by spheres, so they fill it with spheres, and they also fill the ligand with spheres, and then they try to map the spheres onto each other, which should remind you of the putting binding sites onto each other. So yeah, that's the general idea. And so you have the ligand atoms, you have the protein spheres, you make the matches, like here you have points, and you make a graph. You see whether the matches are distance compatible, and you search cliques in the graph. It's completely like superimposing the binding sites. Okay, and at a later time, so this was in the 80s, and later they said, okay, let's extend Dock. We don't want to give up Dock completely. This just works with rigid ligand, rigid protein. I want to go a bit further and make the ligand flexible, but still use Dock. And the idea is to first take an anchor in the ligand, which is still rigid. I place that, and then I start to grow from there. So I dock the anchor, and then I attach other stuff. Then I look for can I attach even more, take out those that have a good energy, and optimize. And if I have other anchors, I start from the beginning and try to find out those configurations that have a good docking score. And because this is, again, a large search base and it can take a while, they also found out that in the beginning it's okay to just do a bump check, just see that the spheres don't overlap, and only later, when you want to find really good configurations, to apply a force field like amber or grid. Another method which is a bit newer is Autodock. And the methods I present are also those that you can use more or less freely. Autodock and Dock are not only commercial, because there are loads of tools out there also that are commercial, because it's closer to success in the value chain for pharma companies, so you can get money from that. In bioinformatics, a lot of the tools you use are free. In chemoinformatics, that is not true anymore. Okay, so Autodock has an empirical scoring function. So the first one I showed you had the force field, and now this one has an empirical scoring function. It starts from any random location and orientation, and then from there it moves the molecule, and it tries out translation, rotation, and also some conformation changes. And now you can explore the different possibilities of changing this and of doing optimizations. The algorithms that are in there is one simulated annealing. I guess you probably heard about all these algorithms before. If not, then ask. So the acceptance of a new state which you have generated depends on its energy, and the more optimal the energy, the higher is the value to accept it. But even disfavorable energy changes are accepted in order to be able to go over an energy barrier. Then you can have a genetic algorithm. There the different movements and changes are encoded in the bit string. And you have a number of bit string, random ones in the beginning, and then you score them, and now you recombine them, you score them, and the fittest ones are taken to the next round, and then you mutate again and recombine, etc. What I find funny is that in the computer we also have Lamarckian evolution. Although I recently learned that on the molecular level we also might have some Lamarckian evolution in biology. So the general paradigm was always that our genes are fixed and we can only pass on what is in there. But it looks like we have some of adaptation which we can also pass on. Yeah. Yeah. Okay, but anyway, so in the computer you can of course also have Lamarckian evolution, which means that before the next generation is made, you optimize a certain pose and then you recombine and mutate, etc. Okay, so that was Autodock, and now Flex, FlexX. Goes to a lot of trouble to represent the inner torsion angles very carefully. There is a very detailed analysis of what kind of interactions do different kinds of groups make. So a very detailed description of where the angles of H1s around something are. And this makes it possible to have a rather precise interaction energy geometry. It uses an empirical scoring function, and at a later point it also started to use one of the scoring functions I showed before. Okay. the way the ligand is put in there is you first select the base fragment. And the base fragment is one that makes as many interactions as possible to make that rather specific. So you have a large search space, and you try to start with the thing that is most defined. So if you look at the ligand, you see that, for example, here you've got possibilities for directional interactions in all those H bonds, and therefore this makes a good fragment. And it's also, and you take a rigid thing. Then you place the base fragment, basically like always before, you try to find matches between the points. You score that, and from there you do a tree search where you extend the ligand, and in order again to be able to explore as little of the tree as possible, you first take the directional interactions because they give you a more defined geometry. Okay, so now I've shown you a number of methods, and if you're given the task, dock something for me please, you have to choose, what do I take? And of course there are also other people who have the same question and who make comparisons. However, most comparisons are done when somebody tries to present a new method. And of course, if you want to get your paper published, you have to test the method in the way that your method comes out good. So it's always a bit tricky to really know which comparison to trust. Or the other way around, I mean, I don't think that most of them forge something, but if you have a task at hand, it's good to find the comparison that's kind of trying to solve the same problem as you're solving. And it's really, as you will see, it's really hard to say something general about docking methods. You can't say this one is good or that one is good. Okay. How did you evaluate in the first place? Well, one thing you can do is to try and reproduce the crystallized conformation. So you take out, you have a protein which was co-crystallized with something, you take out the ligand and you see, can I get it back in the same conformation? The other thing is you can say, okay, I have several ligands and I want to order them in the same way as I would order them by looking at the binding constants. And the last thing is I say, okay, I have a whole bunch of structures and I just want to get a higher proportion. So I want to take out some of the structures and get a higher proportion of active ones in the subset than in the overall set. This is called enrichment factor. Okay, just to give you a feeling for the values, I'm showing one example of what this looks like. So in green, you see the ligand in the co-crystallized form. In blue, you see the docked conformation that was on the first rank. And in yellow, you see the one that comes closest to the crystallized ligand. So the RMSD you would get for the yellow one would be on between 1 and 2, closer to 1, and the RMSD you get for the blue one would be between 2 and 3, so 2.5 roughly. Just to give you an idea, how close does this match and how well can I see interaction, etc. at certain RMSD values. Now, a comparison of loads of different docking methods. I'm showing you that comparison because it's got a high number of docking methods mentioned. The critical thing one could say about this comparison is that they put in a certain way of making up the conformation. And they wanted to be fair, and give the same chance of finding a good conformation to every docking method, but on the other hand, a docking method like FlexX usually builds up a conformation by itself. So giving it pre-calculated conformations is not really fair to FlexX, for example. So this is just to say, whenever you look at a comparison, you have to be really critical of what they do. But still, this gives you a general idea of the order of magnitudes you get. And here, if you look at the Topscorers solutions for the compounds they screened, then the mean RMSDs are between 2 and 4. And you get a solution at all for between 35 and 60% of the structures. Now, if you say, I'm not just interested in the thing that was scored best, but that is actually best, if I really look at the conformation, you also get below 2 angstrom. And you see that the different methods, so how good they perform, also depends on what kind of conformations I give it. But interestingly, this redocking thing is the conformation that is found in the crystal structure, so you don't change anything about the ligand conformation at all, and just give it to the docking program and ask it to put it into the binding site. And this is not always the top performance of the docking method. So in this example of Surflex, for example, it's better if you give it some other ways of generating a conformation. And this is probably due to some clashes and how the docking method says, okay, this fits in here or not. Okay, so that was redocking. How good are we in that? Now the question is, how good are we in ranking different things with respect to each other and in reproducing binding energies? And here you see the different types of correlation coefficient between what the docking method predicts and the reality, and one for the top scoring one and one for the actual best pose. And you see in all cases that we have a positive correlation, but often it's not very high. You have to say though that if you would correlate different groups calculating or measuring the same binding, so the same protein binding to the same ligand, and you would correlate that, you wouldn't get too close to 1 either. Because it depends heavily on exactly those conditions you measure under. So this is also a difficulty in evaluating the docking tools, that the experiments you try to test against are not too reliable or too much dependent on the exact conditions. Okay, the last thing that we want from a docking program is that it's able to extract those ligands that are potent from a whole database. And in this comparison, they took different tools and tried to extract subsets of the database. And they divided that by different types of receptor. And the interesting point is that the enrichment you see not only depends on the docking method you choose, but also it's different for different types of protein you're dealing with. So, for example, in this ACE, Clyde was a lot better than Surflex, whereas when you looked at HIV, they were more or less as well as each other. However, they all do show an enrichment, so it is still better to first look computationally and then only take part of the ligands you could choose in order to really test them in an experiment. So you can still reduce your overhead. Okay, one example where docking was applied, and we always love to show success examples of the methods. Here the idea was to look for good inhibitors of HIV and to also at the same time solve the problem that mutations of HIV can often make your inhibitors useless. Therefore they took a bunch of HIV, so they had one target protein, and for that they took a bunch of sequences with different mutations, and they docked against all the structures. And they took a library of 13 million compounds and docked against the wild type and the mutant structures. And then they looked at which molecules are top scorers in all of them, like not just the wild type but also mutants. For those they did a more careful docking, a more careful scoring, and after that they visually looked at 1,500 structures. Because computational chemists, after a while of being in that job, develop a feeling that hasn't been encoded in any scoring program yet. And they selected 9 compounds they actually tested. Among those 9 compounds were 3 that bound in the micromolecular range. Micromolecular is not great for a drug, but at least it's a good starting point where you can start to optimize from. Okay. And in the case which I showed you before, with the looking for similar binding sites, they also use docking to evaluate whether the thing they found could really be responsible for side effects, because they also took molecules that bind to ER alpha and docked them against the other receptor they found, and then looked at do we see correlations in the docking scores. And they said, okay, we also see correlations there, therefore we think this is a reason for side effects. Okay, so that brings me to the end of the lecture. The take-home message on docking is we can find correct structures in about 70% of the test cases, so find a good docking pose. We cannot always identify that. Ranking the different geometries is good, but it's far from perfect. Ranking different ligands, which with respect to each other, there we can see correlations, but they're not too strong. And really estimating the absolute binding constant is more or less unsolved. And the main challenges that probably stand behind all these problems is that we're not really able to handle protein flexibility because it introduces just too much options. Yeah, we need to be able to improve the reliability of this affinity prediction. But anyway, even though there are certainly problems to solve in docking, it's definitely better than just random guessing, and it helps guide you in the right direction. Turning this the other way around, whenever you're asked to dock something, it means that you need to be able to find a way to validate your result. Either have an experiment or some other kind of prediction method, and the more information you put in, the better. Yeah.
Do you know if some of the methods do like, because you said that the reliability has to be improved, is there also like a
the reliability score?
Like how?
No,
because
not that
I
I know
never saw
of.
that.
No, I don't think there is for.
So the method itself doesn't say, oh, I cannot do this.
No, can't really. So one thing you can do is to see how diverse the different docking poses are, for example, that are put out. Yeah. And whenever you really do that in a real scenario for drug development, you really see that you put in as much information as possible. Often, if you have a first hit, then you try to co-crystallize, and then you have a first idea where it might go, and then you give the docking programs, you place the anchor, so you do it instead of letting the docking program choose and things like that. So the docking helps you explore, but the more information, the better. And there are also, I've once read about a method that tried to find a ligand for an orphan receptor, and it's really hard to do that. And one method or one application was successful, but also only because they restricted their search space enough to be able to really, out of all the molecules that are there, to pick ones that are really likely.